The research paper I'll be discussing today is titled Structural Rearrangements and Selection Promote Phenotypic Evolution in Anolis Lizards by Aria Donoso et al. 2025. This is a recent study, and I found it in the Genome Biology and Evolution journal. The the main idea of why the study was conducted was to understand how genomic characteristics, specifically structural variations in chromosomes and patterns of natural selection, contribute to the high levels of species diversity and dramatic differences in traits. So the phenotypic displays within Anolis lizards. Um, Specifically, Anolis lizards were used because they can be displayed as a classic adaptive radiation model. They're one of the best species to study when studying adaptive radiation. This is because Anolis lizards, there are 400 plus species. They have diverse traits and they're spread across various habitats throughout the world. On the right here, you can actually see the phylogenetic tree that is presented in figure one of the study. You can see how extensive it is, how many branches there are, how many clades, and there are about 400 plus individual species within the Anolis family. So this is a great way to get as much diversity and genetic variation as you can to study adaptive radiation. Um, so these adaptive radiations serve as natural experiments, basically, for exploring how ecological opportunities and genetic factors drive evolution specifically. And in the bottom left, you'll see a picture of an Anolis lizard, which I'm sure many of you have seen before. The ultimate goal of this study was to understand how genome structure helps drive diversity. So, what were the researchers' hypotheses? The authors hypothesized that different forms of genomic variation, including chromosome level rearrangements, and then changes in repeat density near key genes, so transposable elements, and selection on both protein coding and regulatory regions can drive the evolution of distinct traits. So basically, different arrangements of chromosome level parts and how they contribute to distinct traits. They predicted that examining species with different phenotypes would reveal different genomic signatures and candidate genes linked to those unique adaptations, for example, body size, tail length, and cold tolerance. And on the right here, you can see figure one, that phylogenetic tree we saw earlier. And on the right is the genus-wide phenotypic variation in the specific things I talked about, snout length, tail length, and thermoclimatic niche. So that's what this right figure over here is displaying, um, the genus-wide variation. So there's a lot in each one of these traits. So here's a summary of the results. The authors, they generated two new high-quality chromosome level genome assemblies for two different species and then compared them with two other species, Carolinius and Sangri. And major structural rearrangements were found in certain chromosomes of these anole specific particular species. These breakpoints were located near hotspots of developmental genes involved in body shape and growth, specifically axon 2, BMP2, DDIT3, and WIST2. Those are all just developmental genes, and that's where a majority of the breakpoints were found. Across all the studied species, regions with high density of repeat elements were frequently found. And then analysis of protein coding and regulatory regions identify genes and pathways that are likely under selection, and these were often associated with key phenotypic traits. For example, genes under selection were linked to lipid metabolism and mitochondrial function when they were tolerant to cold. So, like I said, detected selection on genes are linked to unique species characteristics. This figure to the right is the number of repeat elements for specific parts of the genome, which ultimately give us those different characteristics. So, why is this important? This research reveals that genomic rearrangements and natural selection combine to kind of foster phenotypic diversity in Anolis lizards, supporting the idea that genome architecture plays a direct role in adaptive radiation. So basically, these findings suggest that evolvable genomes possessing both structure, liability, and adaptive signals help explain why some organism groups become exceptionally diverse and varied in form, like the Anolis lizard.